This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 879, recorded on March 22nd, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's partly cloudy, and it's 4 degrees Celsius and 49 degrees Fahrenheit.
From Western Massachusetts, Alan Dove.
Good to be here, and it is a blustery spring day. Westover Air Force Base is reporting 52 Fahrenheit, 11 Celsius, and wind is northwest at 21, gusting to 28 miles per hour, which is breezy. Is
that not far from you?
Yeah, they're right up the road. They're they used to be a base for B-52s back in the Cold War. So they've got the 9,000-foot runway with the Christmas tree at the end, where they would park the B-52s with the engines running in case they had to go start World War III. Now they're an Air National Guard base for C-5s, which are these enormous cargo planes that go roaring out.
What's the Christmas tree mean?
The taxiway has just branching parking spaces off it.
Ah, okay.
It looks kind of like a Christmas tree.
Isn't it amazing? We used to fly B-52s 24/7, right?
Yeah.
Whose idea was that? Curtis LeMay or something?
It was part of that whole duck and cover era, you know.
My gosh. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It's 58 degrees Fahrenheit, 14 C, and sunny, and also somewhat air-conditioned inside.
Somewhat air-conditioned?
Yes, I'm getting a little chilly. I'm realizing I need to start bringing a sweater.
Is the air on in your place?
I think so, yeah.
Wow.
I'm chilly today.
No, here the heat's on, but I have an old steam radiator here, which I have a handle that I turn on and off when it's too hot or too cold. The thermostat doesn't do anything.
Oh, fond memories of New York. Yeah.
And from Austin, Texas, Rich Condit.
Hi, everybody. 63 degrees, sunny, also quite windy. Though not as windy as last night, if any of you have been paying attention.
Oh, yeah.
News. We had some tornadoes blow through Texas. Man, they got actually aerial footage from drones of these things doing their business, and it's scary. I've never seen pictures quite like this. So some people had the misfortune of being in the way, their houses anyway, and got really torn up. It's amazing.
We have a couple of announcements before we get into our papers today. Kathy, you have a few things to tell us.
Right. So first of all is ASV public service announcement. We want to remind you that there's applications for 2 programs for ASV members to encourage attendance by scientists with families. One is ASV Cares, where you apply for a grant for dependent care. It could be at your home or some relative's home, or you could bring the caregiver to the meeting along with your kids. So that's one. And the second is on-site childcare, which we're having at Madison. It's going to be in the Hilton. They're connected to the Monona Terrace through a third party that's been doing this for years, Kitty Corp. So check it all out on the ASV 2022 homepage. And it's a big red link. And you can get to that homepage just by going to asv.org. And the second one is that I have a postdoc position available in my lab, and I've advertised it on the ASV job site, and I'll tell you more about that when we do our Pics. But you can find that ad and contact me if you're interested in Ann Arbor and my lab.
Cool.
All right. And Rich, what have you have to tell us?
I just want a quick science follow-up from our episode and my pick of the week. Last week, we were talking about the history, sort of evolution of the smallpox vaccine, and about how it's been kind of sort of assumed because of its origins that it was cowpox, and that's turning out to be not the case. And there's a lot of evidence now that at least bits of it or some of it was horsepox. And we were talking about the fact that this is evident to those who are willing to listen or consider it in Jenner's original inquiry. Okay. And I, at the time, as I was describing this last week, said that I recalled doing that episode and recalled reading the inquiry and noting that Jenner was suspicious that the vaccine that was most successful actually probably originated in horses and maybe was passaged through cows. But I, at the time, and the disease in horses was called grease.
The grease, yeah.
Yeah. And I was skeptical. I even, now that I've re-listened to the episode, I even remember looking up grease and discovering that it was or reading that it was a bacterial disease, not a viral disease. And in fact, I went back out of curiosity and listened to that episode, TWiV 170. And we did indeed discuss it. But my comment on that was that Jenner was wrong.
Yes.
And that grease was a bacterial disease. Okay. Now, thinking about that, it's possible, since horsepox basically is gone, that the grease in Jenner's time, or some of it at least, symptomatically looking similar to present-day grease, was in fact viral. One of the reasons I wanted to bring this up is that one of the things pointed out in the Esparza and Damaso article that I mentioned last time is that scientists, like everybody else, are susceptible to myth and groupthink. And it's been very, very difficult to disengage from this notion that it's cowpox.
Yeah.
Mm-hmm.
All right. And I cite myself as an example. It was right there in front of me with Jenner saying he thought this came from cows. And I think, he doesn't know what he's talking about. And there's still resistance, but it's going to turn out to be, that's the way it is. So I think that's one of the most important things.
So now I'm trying to think what the word would be if vaccine comes from Latin for cow. I don't know what Latin
would you be equinated?
You would be. There is a process. You can be equinated.
Oh, okay.
Yes. Yeah.
But is it correct still
It reminds
that
me of something from the movie. Sorry to bother you, but
so he took pustules from a lady who milked cows, right?
Correct.
And she got them from the cow, I presume?
Yes.
Not from a horse.
Correct.
And maybe the cow got it from a horse. That's
the way it's described, yeah.
Okay.
Right.
But,
so you could.
But yes, Jenner was thinking, if you read through this, that he was thinking, because it was not easy. I mean, there's blistering lesions all over the place with all sorts of causes. And it was not easy to obtain something from cows reproducibly that would have the effect that he wanted. Okay. And so sometimes it worked, sometimes it didn't. He thought there was also a seasonal component because there was a time of year when grease was really prevalent in on the farms. And that he thought that grease was actually being picked up by the cows. All right. And that it was that stuff that was most effective in the vaccination. And I'll bet he was right.
yeah. Yeah, and horsepox is fascinating too because we kind of accidentally eradicated it, right? I mean, it's basically gone, but nobody tried to get rid of it.
This is a problem with talking about horsepox. Yes, it's gone. I don't know why it's gone.
Right.
But the only isolate that we have that we call horsepox came from a horse in Mongolia. There was one isolate, and there's no other around that people can discover. So, is that the same stuff that Jenner was playing with? Who knows? Okay.
So maybe we should change it to equination because then the anti-equies would have a hard time pronouncing what they want.
Yeah, there you go.
Right? Because anti-vax just...
The anti-ec movement.
Well, here in New York City, Grease had a long run.
Yes.
Yes.
Yes, it did.
True.
That is the musical. All right. Today we have a snippet in a paper And the snippet is published in Nature Communications, and the title is De Novo Emergence of a Remdesivir-Resistance Mutation During Treatment of Persistent SARS-CoV-2 Infection in an Immunocompromised Patient, a Case Report. This is from a group at Yale as well as Fred Hutchinson Cancer Research Center. We have as co-first authors Shiv Gandhi, Jonathan Klein, Alexander Robertson, and Mario Peña Hernandez. And then we have Albert Coe as the last author. So it's a case report with they actually measure infectious virus. I'm so happy.
Yes.
I just
I
love
was
it.
reading this. Every time I see something like that, I'm like, oh, maybe they listen to TWiV.
It's very good. So this all has to do with remdesivir, which everyone knows is used to treat patients that have COVID. It's given intravenously, so it's a little bit difficult to just get. But there isn't any resistance out there in the wild, that is among patients, although we covered a paper earlier on TWiV that described the selection of resistant variants in the laboratory. That was one from Massimo Palmieri's group at Glasgow, right? That was a very nice paper where they found changes that would confer resistance, but they barely found those changes in all the databases, right?
Yeah.
In all the millions and millions of sequences, very few, and there didn't seem to be any penetration of those. And they concluded that it probably confers a fitness loss. Now, I don't remember. Does anyone, that original paper that we did, which I think was a PLOS pathogens paper, did they look at fitness at all? Did they look at reproduction and make any
I don't think
conclusion?
so.
Yeah.
I don't recall specifically, but I don't think so. And I want to remind the crowd out there that remdesivir is a nucleoside analog, and so targets the actual genome replication process during virus infections. So it is active on actively replicating virus, and it, if you'll recall, it actually gets incorporated into the RNA, but it's got a bulky cyano side group off of the ribose. And so once the polymerase has elongated about 3 nucleotides after incorporating it, this bulky side group runs into a side chain in the polymerase and stalls the polymerase and causes termination.
Great story.
Yeah.
Great story. And that's that episode, TWiV 819, How to Stall a Copy Machine.
Yeah, I love that story. All right, so that change, they looked and they find it, but doesn't look like it propagates and associates with any resistance. So in this paper, they say, well, what about in an immunocompromised patient? Would we see anything different?
Well, they start with the patient.
Yeah.
So it wasn't that they went necessarily looking for this, but they happened upon the natural experiment that I think we were all kind of wondering about.
But they do say in the introduction, selection for resistance is a specific concern for immunocompromised patients who may receive multiple courses, right?
Yeah.
So that's for any kind of infection where you have to give multiple courses. So
they
do have a patient.
They got a patient who has an immunocompromised, a compromised immune system, and who developed an indolent, protracted course of SARS-CoV-2. I just find the word indolent entertaining because in a medical context, it means it's not healing, but in other contexts, it means lazy.
So
indolent is good.
For comparison, how long are patients usually treated with remdesivir?
It's a 5-day course,
5-day course,
as I recall. So this is not too extended. It's only 10 days, I think. Is that right?
Yeah, that's correct. It was a 10-day, but she was in the hospital for a year.
Yeah, they're tracking stuff out to day 170. This happens. Like, oh my gosh.
I'm sorry, not a year, day 51.
Yeah.
And since this acts on replicating virus, and since under normal circumstances the infection is an acute infection, this is one of these things that you have to try and administer early.
Right.
While the virus is replicating. This is a special circumstance because this person has a drawn-out infection.
70-year-old woman. She had non-Hodgkin's lymphoma, was treated for that and had some complications, but it was in remission. So she was treated in 2019. She was in remission for the lymphoma, and then in May of 2020 developed SARS-CoV-2 infection and ends up being hospitalized twice in the following months for that. And she had low PCR CT values, no antibodies detectable, clear lung pathology, right? So the virus is reproducing and causing pathology in her, and ends up in the hospital for 51 days, during which time she continued to have fevers and so forth. But during her hospitalization, she got a 10-day course of remdesivir, and then she seemed to respond to that, decreasing, they say, initial virologic response, decreasing numbers of plaque-forming units.
Yes.
I love it. As well as PCR, of course, which is very nice.
Fever went away.
Fever went away. Then she had a recrudescence. That's another good word, Alan.
Yeah, I like that.
Indolence and recrudescence, where the recrudescence, the infection returns, or maybe it was never gone. It was low, and you didn't know it was gone. It was low. She had viral shedding after therapy, increasing PFU. So she then got 8 grams of monoclonal antibodies. 8 grams.
Yeah.
On day 163, and then
right. So there was the initial hospitalization, and actually this is all open access, so people can go look at the figures we're talking about too. In Figure 1 at the top, they've got things helpfully laid out, their symptoms versus the various hospitalization spells, because she ends up having 3 hospitalizations we're going to talk about.
There's a word that you guys didn't hit on yet, and that's the defervescence.
Yes.
Yes. What does it mean?
It means cooling off or abatement of fever.
The ending of the fever.
Wow. I've not heard that one used.
Yeah. Yes, I noticed that one too.
And by the way, I think that this is relevant. Her SARS-CoV-2 infection happened in May of 2020.
Yes.
Correct.
So this is early during the pandemic.
She's not vaccinated.
No,
she can't.
No one's
it doesn't
vaccinated.
exist. Yeah, no one's vaccinated.
And they point out that, when they first gave as I understand it, the way the paper's written, when they first gave the remdesivir, the monoclonal antibody treatment wasn't available.
Right.
Mm-hmm.
Okay. And it was apparently approved during the time that she was treated, and they swooped in on that and gave her the monoclonal antibody.
Right. And remdesivir was sort of known before all of this for other viral infections, which is why it was sort of the first one they could have tried.
And in fact was developed to be a broad spectrum antiviral. So it's kind of nice to see that that actually worked out.
And as back in May of 2020, I don't think there was all that much known about how effective remdesivir might be for a SARS-CoV-2 infection.
No. That's correct. I mean, it's still, it's dicey because if you don't get it early enough, it doesn't have much of an effect. And that being intravenously given, it's hard sometimes to get it early, right? So,
although in a case like this with an immunocompromised patient, even later on, it's worth taking these antiviral approaches because the virus is still there.
Special case. Daniel says, every update he gives a list of and remdesivir is way at the bottom of the preferred antivirals underneath Paxlovid and monoclonals. It kind of, remdesivir and molnupiravir are both at the bottom, interestingly.
Right. But at the time, remdesivir was kind of the only game in town.
That was it.
Yeah.
That's right. Well, you could take bleach, Rich.
Yeah, that's right.
Oh, yeah, I forgot. And UV light.
Yes.
Anyway, probably hydroxychloroquine, right?
Hydroxychloroquine. Yeah. And then soon after that, ivermectin, right?
Yeah. All the above.
Those were the days.
Still are, unfortunately.
Yeah, sadly.
So the monoclonals really knocked down viral titers. And it also got rid of her anosmia. Isn't that interesting?
Yeah.
17 days after monoclonals. And then she was discharged. But in the meantime, they had lots of samples from her. So they did genome sequencing on nasopharyngeal saliva and stool and blood specimens. They assembled 27 whole SARS-CoV-2 genomes. This is something she could put in her portfolio.
Yeah.
27 genomes from me. I think that's so cool. And an important thing is that clearly all of these viruses were derived from the first virus that she got. They weren't.
Yes.
She didn't get multiple infections, right? So she had one infection, and then it recrudesced, which means
Yes.
it got low, and then it came back up again. And you can tell that from analysis of the sequence.
Yeah, I was glad to see that analysis because, of course, a case that goes on this long, well, how many times has she been infected? Only
Yeah.
once. It's just going.
But of course, they found a change in NSP12, E802D, temporally associated with treatment with remdesivir.
NSP12 being the polymerase, right?
Yes.
That's right. That's
Non-structural protein.
And they did not find this in the first 5 days of therapy, but only after the 5 days of remdesivir therapy. And eventually it increased in frequency to almost, in one case, 96% in saliva specimens. Interestingly, it was 23% in nasopharyngeal and 96% in saliva specimens. So it's different. They talk later about compartmentalization of various viruses might account for that.
Yeah, it's also.
But they also have other changes.
I was just going to say, it's also interesting that it didn't really emerge until 7 days after they started therapy. And so this might be, something that you see somewhat more rarely in people who are treated with a 5-day course.
Yep. And so this change is in this palm of the polymerase. And it could be analogous to the change is this the same position as the one that we...
No.
Is it different? What was the one in that paper?
I'm just now looking it up. I had looked it up before, and I will tell you in a moment. Serine 861.
Okay.
That's the one that causes the steric clash. And the way they describe this residue is that it's part of a cluster of amino acids that lend to the structure of the active site, put it that way. Okay. And so they're thinking that they don't really have a precise mechanism for this other than it relaxes or alters the structure of the active site in such a way that steric clash is no longer as important. Okay. But this makes sense in terms of what else they find. I'm getting ahead of myself here, but this is interesting because the virus doesn't replicate as well. It's not as fit.
Yeah.
So there's a fitness cost for the mutation.
My read is that maybe there is less tight binding to the RNA because it does mention that we're messing with the area that stabilizes the loops involved in binding. So perhaps the polymerase can't really stay on the RNA too well.
Yeah. Yeah, it actually, I mean, they're obviously, you'd have to look at this with detailed structural studies, but the model kind of holds together. You've got this in the palm of the active site, and we know that you jam that up with remdesivir, and now this is maybe relaxing it a little bit and letting the remdesivir not interfere.
So they introduced this amino acid change on its own. They actually put two changes, E802D and E802A, which was part of the previous study. So I don't know where that 802 comes from, Rich. You said 860, but what is this? Did they find 802 also in that previous PLOS pathogens?
I don't recall. I'm just
look at that.
I'm sitting here looking at the movie of the steric clash. Remember?
Yeah, that's pretty cool.
It's just awesome.
Is the PLOS pathogens paper Semiel, Szemiel, because they cite where 802 as previous information came from, and it is a PLOS pathogens paper.
No, the mechanistic paper that we looked at before
is Nature
is Kokic, Kokic,
right?
First author, last author Kramer, and it's a Nature Communications paper. So the 802 is something else.
Okay.
So am I remembering correctly, there was a PLOS Pathogens paper where they selected in vitro for resistance, right?
Yes,
In cells
and that
in the
is
laboratory.
reference number 2 here. Is in vitro selection
Okay, and what's the residue?
remdesivir 802.
Is it also 802?
Yeah.
Yes.
That's
Okay. Got it. I think we did those in separate episodes. Yeah, we did one with the stalling movie with the structure and the stalling, and then separately the resistance. Yeah. Okay, so they introduced these 2 amino acid changes at 802 into an infectious clone, so it's just that change. And in fact, the viruses don't reproduce as well with those changes. They're less fit. They say these changes may impart a fitness cost. And when they add remdesivir, the 802D reproduces to higher titers than parental virus, so confirms that it confers resistance. They also do dose response curves, which you should do always, and you can titrate the resistance, and you can see that clearly this confers resistance. So that proves that the change does that, which is important to do. We speculate that the changes distort the active site in a way that it either enables the enzyme to exclude the drug or alleviates the steric clash mediated by serine 861. So that's the position, Rich, where they found the clash in the movie, I guess, right?
Yes.
Yeah. All right, and then they say probably the fitness cost is why this doesn't propagate in people,
yeah.
right, in patients. They've been found. So out of the 4.8 million genome sequences from patients in the database, these 2 changes have been found in 131 and 290 patients, but there's no evidence that they spread. So there you have it. That's and in fact, with this, these isolates, they say there are 6 changes. Actually, we're only talking about the polymerase change, but there are other changes, which is, a common observation now that we're seeing in immunocompromised patients where you have long-term reproduction. You have a lot of changes accumulating, which you may remember we talked about in the context of Omicron emergence.
So one of the things I liked about this was that when they quit the remdesivir treatment, in at least one compartment, the mutant virus disappeared.
Yes.
Consistent with there being a fitness cost. Okay. But the other thing that's puzzling, I think, even to them, is this idea of compartments. All right.
Yeah.
Yes.
Yeah.
Because the mutant disappeared from the nasopharyngeal specimens specifically, but I guess not from the other compartment was, I don't understand how nasopharyngeal compartment can be all that much different than saliva.
Yeah.
Yes.
Okay. But
I agree.
but there it is. And they don't really have an explanation for that.
No.
So the other thing that I wanted to ask Brianne about is the immune status, if you know it, of, in some at least modest detail, of a patient of this sort. Okay, because they gave me the impression that this person is significantly compromised in their B cell response, but it's not clear where the T cell response is. And it's interesting under those circumstances that they're not dead from this infection, okay, but having trouble controlling it. So can you bring any perspective to that?
So they
Brianne?
talk about the fact that this patient was on rituximab for their non-Hodgkin's lymphoma. And so that indicates to me that they have a tumor of B cells where they have excess B cells. And they're being treated
So
with
rituximab is what?
Rituximab is an antibody against CD20 that's on the surface of mature B cells. And so it's killing mature B cells, which includes the tumors, but also all the other mature B cells in the patient.
Okay.
So they also
So you're seriously B cell deficient.
You are seriously B cell deficient. Yes. And so usually patients like this might end up getting something like IVIG. So they're getting antibodies supplemented in. They mentioned another drug
they mentioned another drug I don't know about here as well. Yes, that was if I looked this other one up.
Is this bendamustine?
Yeah, hang on. Let me. Okay. Maybe we're talking about the one I looked up was filgrastim.
Okay.
Filgrastim, which is it's granulocyte colony stimulating factor.
Okay.
But maybe there was another drug as well. At any rate,
Earlier
so she's
on,
B cell
they talk
deficient.
about another drug, yeah.
Okay.
So, Brianne, her T cells would be okay, you think?
Her T cells would be okay. They would, miss out a little bit on having some antigens presented by those B cells, but in theory, the T cells should be okay.
Of course, that's why she survived.
Exactly.
They have this sentence in here. It says, given reports of maladaptive immune responses contribute to the risk of severe COVID-19, we speculate that the compromised immune response was unable to control the viral load, yet conversely protected the patient from developing severe complications.
Yeah.
Potentially,
I bet T cells had some role in that, Kathy, here's another word for you. Blood dyscrasias. Do you know what that means?
I didn't see that. Didn't look it up.
Dyscrasia. All right, we got to look that up. Dyscrasia. Dyscrasia is, in my wonderful internet here at the incubator, an abnormal or disordered state of the body. Various blood dyscrasia. Okay. Abnormal condition or disease of the blood. That's really
how I feel most of the time. Dyscrazy.
Dyscrazy, yes.
And resolved. So apparently, why do they say in the setting of a negligible cellular immune response? You looked at the T cell response. Anyway, the monoclonal antibody cleared viral shedding, abrogated anosmia, and resolved the blood dyscrasias. Okay. So she had some abnormalities in the blood. That's a new one for me.
Okay, the history and etymology, Middle English, diseased condition due to disproportionate mixture of the humors.
Ooh.
So it's actually borrowed from Greek. Dys and crasis is mixing, blending, temperament. And there's another one here, to mix, mingle as wine with water. Karyenyi, erni, me.
This is a new word, rich paper. It's good.
Yes.
Yeah.
By the way, Brian, they say that this person had evidence of T cell exhaustion, right?
Which could potentially, I could see.
Yeah, so maybe that's why they say cellular immune deficiency.
Right. I mean, if we're looking, go ahead, Rich.
I'm wondering what T cell exhaustion is.
So in the case of chronic...
somebody who's tired of hearing about them.
Brianne's students get this sometimes.
I have a great demo for this with my students that may involve laying on a table as if it's a hammock. But basically, if we see chronic infection and T cells are seeing antigen for really long periods of time, then the T cells can start to lose their functionality and stop making cytokines, stop being cytotoxic, and even go to apoptosis sometimes. And that's called T cell exhaustion. It's also kind of important in responses to cancer and in autoimmunity because those antigens are not going away anytime soon.
And that's reversed by checkpoint therapy,
Exactly.
right? Like anti-PD-1, because PD-1 is programmed death 1, which is one of the proteins that's doing that apoptosis.
Yes,
right?
and it's PD-1 was sort of an important protein at the beginning of the T cell exhaustion literature because it was the protein that you could use to find the exhausted T cells.
Right. What's the other one now? There's a second one, right?
A second one for exhausted T cells, yes, and I don't remember what it is right now.
It's one. They also use that for checkpoint therapy.
Well, they also use CTLA-4 for checkpoint therapy.
CTLA-4,
but that's
right?
not really an exhaustion marker.
I learned so much from this stuff, and it's all on the fly, right? You know.
Hey, who knew you could learn from conversations?
Amazing.
Yeah. Anyway, they end up by saying we should pay attention to the genomes in immunocompromised patients. And I would agree. It's a good idea. Okay.
I wonder if this is if there are other examples of, because, there's this, one of the hypotheses to explain the evolution of Omicron is that it simmered in a chronically infected, perhaps immunocompromised patient for a long time and then came out. Okay. I wonder if there are other examples of virus SARS-CoV-2 actually evolving in a patient who has a chronic infection. It must be.
I think there have been other studies that have looked at virus longitudinally in some of these patients. I'm not sure that they've been able to assign changes in the phenotype as the virus changed, but I think they've looked at sequence changes over time.
If I remember, it wasn't the first paper from Adam Loring a long time ago looking at that first immunocompromised patient, which I think was at University of Michigan, and we did that, and it said there's a patient who had long-term infection and has quite a number of mutations, was unexpected.
All right.
And I think that was the first.
Okay.
And so there have been several since then, and we should be looking more. Yeah. All right. So our paper is an article in Nature, and speaking of exhaustion, the author list exhausts me.
Yes. Well, there's which author list? So there's the one at the top of the paper in the normal location, which has several consortia listed in it among the individual authors. And then there's a little footnote saying that a list of authors and their affiliations appears at the end of the paper. And you can actually go into the full list because it wouldn't fit up top.
It takes up almost a whole page.
Yes, of very small print.
So a lot of people, the name of the paper is Altered TMPRSS2, TMPRSS2 usage by SARS-CoV-2, Omicron impacts infectivity and fusogenicity. And I was interested in this because, well, I'll tell you what the bottom line is, okay? Because they say the last sentence of the discussion is, experience with Omicron has demonstrated that predictions regarding replication and tropism based on sequence alone can be misleading.
Yes.
A detailed understanding of the phenotypic impact of a complex set of mutations is vital. And I agree, and that's what this paper is. They do some experiments in cells in culture to figure out what all these substitutions, amino acid substitutions and spike, are doing. Some of them we've already touched on, but it's okay to be repetitious.
Yeah. They do a bunch of experiments.
Do a bunch of experiments.
And there are 7 first authors.
No.
And Cambridge Institute of Therapeutic Immunology and Infectious Disease, and also some affiliations with the University of Cambridge. And then, as you can imagine, a whole lot of other places.
Yeah.
I like this one consortium, the Genotype to Phenotype Japan Consortium.
Yes.
Mm-hmm.
It's an interesting G2P, G2P Japan. So as you know, Omicron has over 30 amino acid changes in the spike and 20 changes alone in the N-terminal domain and the receptor binding domain. It has 6 changes that were not previously found in other variants of concern. And so the question is, what can we learn about this if we study the virus in cell culture and compare it to like Delta and Alpha, for example? And they say we're looking at the biological properties of Omicron. And these are the kinds of studies you need to do to get some insight on what's happening in people. One of the and this is it's a confusing virus because we did a paper on TWiV a while ago showing clearly that in hamsters it is less virulent compared to, say, Delta. Yet every week Daniel Griffin says this virus is killing patients, and it's killed more young kids than Delta did. 5-fold. There was a MMWR last week saying 5-fold higher, not killing, a 5-fold higher hospitalization rate in very young kids compared with Delta. So it's very hard to make these kinds of conclusions in people, especially if you have a mixture of ages and susceptibilities and immune status and so forth.
Well,
so
and there's also an order of magnitude or more prevalence difference
Yes.
Omicron, I mean, even accounting for widespread testing or testing differences early versus late in the pandemic, this thing spread everywhere and just in quantities that were not seen for any previous strains.
Yeah.
And so,
Right. And,
variants.
and even with the other variants, if you look at, say, when we were watching Delta spread through the population, or when we were looking at Alpha or whichever, there are so many population level differences and differences in mitigation measures, in addition to vaccination coverage. And there's so many ways that it's really hard to compare different variants in people in the studies.
And also, not to put too fine a point on this, but a lot of the people who might have died of this infection are already dead. I mean, we have lost not a small number of people who succumbed to earlier rounds of COVID-19. So we're seeing.
No, it's true. The people now being infected are either immune or they're younger. The comorbidities, as you say, many of them are gone. Yeah. So
and, the vaccines plus the infections of previous rounds, the pre-existing immunity is much higher. So it's occurring in a completely different context from earlier waves.
So one of the things that I like about this paper, or one of the things that it does to me, is that it sort of puts in perspective the difference between sort of epidemiological studies and laboratory mechanistic studies. And this has been, I think, an ongoing conversation on this podcast for a long time. And I think the bottom line is that you need both the epidemiology and the mechanistic studies. Neither one alone tells you exactly what's going on.
Right.
And I like they have a nice way of phrasing the place of Omicron epidemiologically. In their introduction, they say, Omicron has been associated with very rapid increases in case numbers.
Yes.
How carefully done is that? No mention of transmission or anything like that.
No, it's good.
Associated with very rapid increases in case numbers, and recent data demonstrate considerable reinfection and vaccine, in quotes, breakthrough.
Right.
Probably due to evasion of neutralizing antibody responses. And recent findings suggest reduced severity, but they're very carefully worded, avoiding words that
trigger TWiV.
Yes,
that's true.
Right.
Yes, it's true. Nice that they have.
Or at least some of the authors are listeners.
Has been associated and not leads to.
Yes.
Yeah.
I think
they are sticking to the data is what's happening.
Well, I think that's it. I don't think they listen to us, but we are basically looking at the data, and so are they, and you come to to similar conclusions, right?
One thing I wanted to say I hope this doesn't take us off into a tangent, but
that's okay.
Rich said something that to me, this paper is in a way more of a descriptive paper. And, people can really cast aspersions on descriptive research, but I've always maintained that you have to understand and describe the system if you want to be able to then manipulate it and test hypotheses. But if you look at a large proportion of what they're doing here is sort of looking at things and seeing what is the case. I mean, they do some things, they make pseudotype virus and they tested some of those and they use some inhibitors and so forth. But overall, to me, it's more of a, here's all these mutations, here's what we can look at in Omicron and what do we see?
Although from time to time they do use the H word.
Yes.
We hypothesize that.
Yeah.
Right. I would agree. But it does no casting aspersions. I think it needs to be done.
It's great description. It has to be done. Yeah, absolutely. And
especially when it is done in this extraordinary level of detail. There's a lot of craft that went into this paper, and I appreciate that. And for those for listeners who want to look it up, it is open access. But as we've said in the discussion some years ago on how to read a scientific paper, this is not a magazine article you flip through and get the gist in 20 minutes. You're gonna sit down and bond with this thing for a while.
Yeah.
So they go through a number of aspects of Omicron biology. The first is that there are all these changes in spike. Does that affect binding to ACE2? So they do two approaches. They do biolayer interferometry, where they can look at the protein spike binding to ACE2 in a very quantitative fashion. They find threefold enhanced affinity for ACE2 compared to the original and delta receptor binding domains. And they say that's because of a change in dissociation rate. And they also confirm that using cell-based experiments where they have cells producing the protein and looking at binding to them. And so they say this binding could be a factor and now they talk about in the enhanced transmissibility. And I would say, who knows what threefold does?
Right.
And we have actually done a paper where they say the enhanced transmission. So I was thinking about this, and tell me what you think. Now, maybe I'm being pedantic. I think there's a big difference between transmission and transmissibility, right? Transmission is the movement of the virus in the population. And when you say enhanced transmissibility, to me anyway, you're saying that the virus has got something different about it. And that's where I have the issue. So the Danish study
about
we
it
did,
in the actual transfer between hosts.
Exactly.
Right.
So in the Danish paper we studied, they concluded that it was actually the antibody evasion properties of Omicron that caused it to transmit better in households anyway.
Right.
So here the binding, I'm not sure that has any role. It's there, but who knows, right? What is threefold with respect to binding?
Well, as Kathy said, it's a descriptive paper.
It's a descriptive paper.
Now we have this part of the description of it that
maybe
could be
we'll
understand sometime. Yeah.
I think could be a factor is fine. All right, then they ask, what about antibodies? They look at two of the combinations from Regeneron, Casirivimab, and Imdevimab.
And those are the ones the patient in the
and,
previous paper were treated with.
Yeah.
Yes.
That's correct. And of course, that patient was before Omicron, fortunately for her. But the Imdevimab doesn't affect virus infectivity. So that doesn't work. And then they look at individual component monoclonals. And here they compare Delta with Omicron. And they find that the combinations were good against Delta, but complete loss of neutralizing activity against Omicron, either alone or in combination.
And that's really striking.
Yeah.
Yeah. Which we've talked about before, I believe. And finally, remdesivir and molnupiravir have similar activities against Omicron and Delta, so they still work.
Right. So the direct acting antivirals don't seem to be affected, but Omicron seems to have the ability to evade these monoclonals.
And these were done using SARS-CoV-2 viruses.
Right.
It's a mix of viruses and SARS-CoV-2 and pseudotype viruses, which I suspect is when different groups, do their own thing in a big consortium like this, right? Next, they ask what about vaccine-induced antibodies? And this is certainly something we've talked about before. They use both pseudotype viruses and infectious viruses. They have samples from 40 people who were vaccinated with either BioNTech or ChAdOx. And they have samples 1 month after dose 2 and 6 months after dose 2 and 1 month after dose 3. So a nice mixture of different immunizations. Tenfold loss of neutralization against Omicron after the second dose compared with Delta, and that's something we talked about before also. And they say Omicron neutralization was not detectable for most individuals who got 2 doses of ChAdOx. And then neutralization went up after the third dose, which they say suggests an increase in breadth as well as titer of antibodies. And that was actually the Ontario study that we talked about months ago, which convinced me that it was a good idea to get a boost.
Yes.
Yeah, as a matter, there have been several different studies now that have exactly the same result on this. Okay, and to me, this is remarkable that you get not only the increase in the quantitative increase, but apparently the increase in breadth.
Right.
Yeah, I remember
And as I
being
said, they
shocked back when Paul and Theodora showed us those data.
Yeah.
So they also did this with infectious virus and got similar results. And then they also tested sera from people vaccinated with the Moderna vaccine.
Well, can I just back up and ask a question, a broader question here? So there are a lot of vaccines that are a 3-dose series, right?
I don't know how many are there. I don't know what a lot is. There are at least. I just know
I can think of a couple, and I feel like
I think many of our childhood vaccines are 3-dose series.
Yeah.
Oh, you're talking about other vaccines, not COVID
vaccines. No.
Okay.
Right.
Yeah. Sorry.
No,
yes.
so 3-dose series is a.
Yeah.
That's a common thing, and I'm wondering if anybody looked with any of those. I mean, because 3, anytime humans do something, we're like, oh, make 3 of it. So my thinking was that was why we get 3 doses of these vaccines, because, same reason you used to change your oil at 3,000 miles, or there are 3 sections in the plot.
That's to make money for the oil company.
Right. No. So that's just that seemed like a convenient number. And now I'm wondering, has anybody done? Probably not, because by the time you would have had the tools to do these studies, those vaccines were already standardized. But a study where you look at somebody who got 2 doses of measles vaccine and look at their antibody response versus 3 doses, or
so in one of the previous conversations with Shane Crotty, he indicated that you get a qualitative difference in the response after a 3rd exposure to antigen. I actually spent some time emailing with him offline about that afterwards, because I found that really interesting. And it's sort of, you have to kind of pull together some things from a number of different papers. There's not kind of one clear study. It's more of an impression from a lot of places. But that was sort of something that he had said and something that I've been looking at since then.
Okay. So this is a thing, but it's maybe we didn't realize it until now, because now we finally had the opportunity to look at this exact phenomenon.
Right.
That the immune system after the 3rd dose gets broader than it was maybe after the first 2.
I don't know if you could generalize it to all, but
no, but it's
right.
It struck me that, oh wow, that's a neat thing that happened after the 3rd dose, and then I thought, well, is that something that happens after other 3rd doses? And no, you can't generalize because we don't have the data. But as Brianne said, apparently there are kind of hints of this in other literature.
But you wouldn't. I mean, for an infection, you wouldn't expect you to need 3 infections to do anything, right?
No.
But
so
some of the attenuated vaccines are 2 or 3 doses,
right?
Maybe because they're a little fitness compromised and they don't reproduce.
Yeah, but you could
the
also,
other issue.
Oh, go ahead. Rich.
Go ahead.
Oh, I was just going to say.
No, you go ahead.
I think the one other piece is that if you are exposed to something 3 times, that would evolutionarily be something that you probably need to be really on the lookout for and making a good response to, because it's more common in your environment.
Right.
The other issue, it seems to me, with respect to these vaccination protocols is not just the number of doses, but the spacing.
Yes.
Because the first 2 doses are pretty close together.
Yes.
The 3rd dose is spaced further apart. And that, from what we've discussed, strikes me as a different circumstance. And it has sort of historical roots relative to the SARS-CoV-2 vaccine.
Right.
We knew a boost would do something. We needed to get it done as soon as possible. But it turns out that if you then wait a while and give a 3rd dose, you get a different shape of immune response.
Yeah, I saw a study recently where they had people who got 2 doses 6 months apart, and they have the same broad, high titer antibodies as the boosted people.
Okay.
So, whereas, who was it, Brianne, that we spoke with, Gabriel Victoria, said, no, it's 2 primes and a boost. That's how it works.
That's right.
He didn't agree with that, but that's fine.
Well, I mean, and if that's the case, that you get 1 shot and then wait 6 months, and the 2nd shot brings you up to where the 3-shot protocol would have done. I don't want to do that because I want that initial boost.
Yeah, that makes sense.
Yeah.
You know?
you're going to be at risk for those 6
exactly.
months.
Yeah, exactly.
Sure. Absolutely. Anyway, so the Moderna also had a reduction in neutralization against Omicron. They say CoronaVac had no neutralization against Delta and no neutralization against Omicron. So that's the inactivated vaccine made by Sinovac, I believe.
Right.
And
which I was into when they were developing it, and it turns out it's kind of a dud.
But it turns out that they did a test where they boosted them with mRNA, and that works very well.
Okay.
And this is interesting because, President Xi has been trashing the mRNA vaccines, right? He's like, no, our inactivated vaccines are great. And now they actually need a boost.
They need a boost with the mRNA.
To make it really good.
Well, this leads me to this. I've been watching, as I'm sure we all have, this sort of resurgence of disease in a number of locales globally, in particular with the new Omicron variant BA2, and it's showing up like gangbusters in Hong Kong,
right?
And it's showing up in China and elsewhere. And I think a lot of it, I don't know what it's going to do in the U.S., but it seems to me that a lot of this has to do with sort of the immune status of the population that it's moving into. Okay, and apparently the vaccine program in Hong Kong was not as successsual as elsewhere.
Correct.
And
That's correct.
The population is pretty susceptible, and the Chinese population is immunized with CoronaVac.
That's right.
Right.
That's right. That's exactly right.
The other issue, and the reason we're hearing so much about this, is that those countries have taken a different approach to the point that they are still trying to be SARS-CoV-2 free, and so China locked everything down again, and so you don't have the ongoing exposure, and you have a lot of news coverage of this because when China locks everything down, that's a whole lot of people, and it shuts down global supply chains. And similar thing in Hong Kong. So they're taking a very different approach that made a lot of sense early in the pandemic and may not make quite as much sense at this point.
All right. Next, they do a series of nice experiments to understand the reproduction of this virus with respect to cleavage of spikes. So as you know, the spike protein has to be processed by proteases for maximal infectivity. And there is a cleavage that can be carried out by either cell surface TMPRSS2 or by an endosomal protease. And then having a furin cleavage site enhances that cleavage. And this virus has changes around the furin cleavage site that seem to reduce its overall cleavage of the spike. So they want to know what role that plays in reproduction. And part of what they use are primary human nasal epithelial air liquid interface cultures. Perfect.
Yes.
So it's primary. It's the right cell to do. They put it in an air liquid interface, so you have all the cells differentiated into mucus producing cells, epithelial cells, ciliated cells, and so forth. And then they can infect them.
It's a nose on a dish.
A nose on a dish. It's good.
They have a nice little cartoon of how this is done.
Go ahead, Kathy.
Is the furin cleavage site synonymous with polybasic cleavage site?
Yes.
Okay, because they seem to use them interchangeably,
Yes,
and
they do.
I've never been totally clear.
They do.
Okay.
It's funny that you would switch back and forth and not be standard, right?
If you have a gazillion authors, maybe this is one of the things that creeps into the manuscript.
Right.
So the furin or polybasic is quite close to the TMPRSS cathepsin cleavage site, which gives you S1 and S2, right? All right, so in these nasal epithelial cultures, Omicron and Delta reproduce similarly. Doesn't seem to be any difference, okay? Next, a lung cancer cell line, KLU cells. And these cells have TMPRSS on the cell surface. There they see greater replication for Delta compared with Omicron. Then they infect a colon cancer cell line called KCO2. These cells have high levels of TMPRSS, and again, Delta reproduces better than Omicron. And finally, then they take HeLa cells and they got ACE2 in them, the receptor, and they're also producing TMPRSS2. similar results. So the presence of TMPRSS seems to correlate with less Omicron reproduction compared to Delta.
Right.
So then they say maybe this has something to do with spike, either incorporation of the spike into the virus or cleavage of the spike. Delta spike is highly cleaved, they say. So they purify Omicron virus particles from separate infections. They see similar amounts of spike in Delta and Omicron particles in cells that have TMPRSS in them. But there's reduced cleavage of the spike in Omicron compared with Delta.
Yeah. One thing sorry, go ahead.
It's okay, go ahead.
I was just a
Not
little
important.
bit uncertain about the air-liquid interface cultures because they don't tell us what's going on with TMPRSS in those cultures. So do they have it but have lower levels? Do we assume that there's just less of it in terms of density because the cells are heterogeneous? You know, they don't really mention what's up with that. And so I would have liked to know, because I am not an expert on TMPRSS by any stretch of imagination.
They don't mention it. It's important because obviously that is a site of SARS-CoV-2 reproduction, right? Nasal epithelium. it's a key place.
And Omicron does well in it in this experiment.
Right.
And so I assume there is TMPRSS there, but why it would be why Omicron and Delta would be the same? I mean, their whole idea, which they will get to prove, is that TMPRSS is a problem for Omicron, right? But what about these? They never get back to those. No, not that I remember. Although, it's a good point. Okay, so then they say, so TMPRSS cleaves the spike at the plasma membrane, and the virus could enter there. If there's no TMPRSS, the virus can bind ACE2, it gets endocytosed, and then in the endosome, there are proteases called cathepsins that will then cleave the spike and allow entry there.
Right. So SARS-CoV-2 has two entry mechanisms. There's the one at the surface, and there's the one from the endosome.
And the one at the surface where TMPRSS2 is involved, that is enhanced by the polybasic or the furin cleavage site. Okay. The one in the endosome, less so.
Right. The one at the surface is also important in syncytia formation, where an infected cell fuses with another infected cell, which is an important part of pathogenesis from what we can tell in some cases of COVID-19.
So they use pseudoviruses to look at entry of these various viruses. So they have a wild type, they have Delta, they have Omicron as spikes in these pseudoviruses. They find that what kind of cells are here? I'll get to it. They infect, now in this case, they do air liquid lower airway. These are organoids, actually. These are not air liquid. These are organoids from the lower airway, and also gallbladder organoids. I don't know why gallbladder. Maybe it's a control. I don't know. And the KLU3 lung cells. And you will remember the KLU3s have TMPRSS2. Okay? And they find impaired entry of Omicron. So now we're not looking at the whole cycle. We're just looking at entry. Impaired entry of Omicron in the organoid systems and in KLUs compared to Delta and the original. When they overproduce ACE2 in the absence of TMPRSS, so they have cells, HEC293s have a lot of ACE2 and deleted for TMPRSS, then Delta and Omicron reproduce to similar levels. You know, there's a lot of data here, but take away TMPRSS and now Omicron doesn't have a problem reproducing any longer.
Right.
So then, Brienne, they looked at TMPRSS by looking at the RNA in various cell types, right? Mm-hmm. So they say KLUs and organoids have high levels of TMPRSS. The HeLAs have low levels. The 293, it's deleted, so it has low levels. And I was hoping they would tell us about the nasal epithelium there, but they didn't.
No, they don't. They tell us about the airway organoids, and those seem to have a lot with that. So I think for their argument, they would want to say that the airway organoids are not like the nasal organoids. that they would ideally want the nasal organoids to have very little. But who knows?
It would be so easy to do.
Yeah.
So their idea is that the problem of reproducing with Omicron is because of the presence of TMPRSS. And so they show this very specifically by looking at 293s with and without TMPRSS. And you can see when you have TMPRSS, Omicron has a problem compared to Delta. No difference when you delete TMPRSS. So the TMPRSS route of entry seems to be a problem for Omicron. And they say this is probably because of suboptimal cleavage of the Omicron spike. And I would guess the changes around the furin cleavage site in Omicron are making it less cleaved and therefore doesn't work well with TMPRSS. So then if this is the case, if TMPRSS is not preferred, then the entry route of Omicron should be changed. And you can probe that by using inhibitors. Right? There are inhibitors of TMPRSS and there are inhibitors of cathepsin. And so they do that experiment, and the inhibitor of cathepsin is called E64D. The inhibitor of TMPRSS is called camostat. And the camostat in the cathepsin inhibitor has a greater effect on entry by Omicron compared with Delta.
Right.
And the TMPRSS inhibitor has a greater effect on Delta or Omicron's already not doing well in the presence of TMPRSS. So Omicron basically is shifted towards entry through the endosomes. Doesn't enter as well at the cell surface. That's the idea here.
This is a great example of this principle of coming at a problem from several different angles.
Yes.
And getting the same answer from different angles that really reinforces the answer.
So TMPRSS modulates entry by Delta to a greater extent than Omicron. So they looked at the distribution of TMPRSS and ACE2 in human respiratory cells by single nucleus RNA sequencing from 5 locations in human lungs. Higher expression of TMPRSS in alveolar pneumocytes, 2 different kinds of pneumocytes, and lower in the trachea as we move up, right? Pneumocytes are all the way down there. Alveoli are all the way down. Trachea are higher, but they didn't do Brienne's experiment.
What about the nose?
Or about the nasal epithelium.
Yes.
Single nucleus sequencing of the nasal epithelium. You would think this must be known, Brienne. Maybe
I know. I just don't know.
It's a known thing.
Yeah, I didn't look at the methods to see whether whether they did these sequencing samples themselves or whether this is mining databases.
They said we use single nucleus sequencing from 5 locations.
Okay.
Sequencing data.
Data. Maybe it was, reference 24. Yeah. Okay. 24 is a spatial multi-omics atlas of the human lungs.
That's it.
I like multi-omics.
See, they wouldn't go all the way to the nose there.
Right. Because that was the lungs atlas.
If we just Googled, is
it says see
TMPRSS
next
in the
map
nose?
where you need to go to the nose.
Is TMPRSS2 in the nose? Could that possibly yield anything useful?
The internet.
SARS-CoV-2 are highly expressed in nasal epithelial cells. Oh, here's a Nature Medicine paper. Let's see what they say. Where's TMPRSS? Here, TMPRSS2 have been detected. TMPRSS has been detected in nasal and bronchial epithelium by immunohistochemistry, as well as alveolar type 2 cells, which we just found in this paper as well. And so they say here they looked at different data sets for this. TMPRSS was highly expressed with a broader distribution. Cells from the respiratory tree. Fetal tissues, no, I don't care.
I'm searching PubMed.
I think it's...
I'm sure it's got to be in here somewhere, but
they say it's
not.
expressed. They say it's expressed in nasal epithelium, but they don't say... I would think that it may be lower, right?
Yeah, we want a quantitative
Right.
comparison.
We want a quantitative experiment, and they don't do that here. Anyway, so.
Yeah, my first thought
It's an
is just
interesting
because
question.
those are such heterogeneous cultures that
Yeah,
you're going to have
maybe.
overall lower expression because some cells will probably have it and some cells perhaps won't.
Mm-hmm. And then the last experiment, they asked, does Omicron and Delta, do they fuse differently? Cell-cell fusion, right? And I think we did a paper before which said that Delta spike fuses better, right? And causes...
Right.
Yeah, we previously saw that spikes bearing this proline 681 have higher fusogenic potential. And
So the deal here is that
Yeah.
when you infect a cell, some of the spike that gets made is on the surface of the cell, right?
that's right.
And that can
Yes.
interact with ACE2 on adjacent uninfected cells.
Correct.
And that interaction can cause fusion of those two cells.
Right.
And
And they
that
have
would,
a delightful assay that we've seen
is
before, but
delightful,
it's still
yes.
delightful, where they have a green fluorescent protein that's split into pieces. Okay, neither piece can fluoresce, but if the pieces come together, they can fluoresce. This is an old concept.
It's
but
like those
I think
Ziploc bags, yes.
Yeah.
Put the two together and they make green.
Or beta-lactamase.
Or beta-lactamase, yeah.
That's the one that
Beta-lactamase, yeah,
came to
that's the
mind for me.
classic. Very good.
Mm-hmm.
And so when the cells fuse, they turn green. It's cool.
And by the way, this fusion would allow virus particles which assemble intracellularly to move to another cell, right?
Right.
Yep.
And in theory, antibody couldn't get them.
Right.
So they measure cell-cell fusion. They put, the virus spike on one cell with part of GFP, and then there's the receptor on the other. And they can compare Delta and Omicron. So Delta has increased fusion compared to ancestral spike, but the Omicron didn't fuse very well. Very poor fusion.
Yep.
And they said, well, here, Kathy, we predicted that poor fusion would impair cell-to-cell spread. So it's like a little hypothesis-driven, right?
Yes.
So they actually do an infection of cells, and they put on an overlay, carboxymethylcellulose, which is not as stiff as agar, but it's kind of thick, right? The herpes virologists like to use that for their plaque assays. I don't know why I don't use agar. Maybe it doesn't work in agar. But then they can ask, do we see foci from original cells, which would indicate the spread of viruses by cell-cell fusion, which are basically plaques.
Yeah.
And they look at them under the microscope, and they look at the size of the foci. The numbers are the same, but the size of the Omicron focus is 1.5 times 10 to the 3 square microns, and the Delta is 3.4 times 10 to the 3, 2.3-fold higher. So the spread of virus is poor with Omicron, consistent with the fusion being poor.
They actually
Yeah,
refer
nice.
to this
They have
as small plaque sizes.
Yeah.
Small plaques, yeah.
Yeah.
Small plaques as well, yeah. And so this is probably caused by these changes in the spike. We don't know which ones, right? Because they're 20 or more. So that would be some job to sort out those, I guess.
Yes.
But what's interesting here, and they mention this, so Delta has this P681H change in the spike, which supposedly increases its fusion. Omicron has that, but it also has two other changes, and maybe they knock it back down again. So that would be interesting to sort out. I'm sure somebody is doing it. All right, so those are the experiments. So we have the spike doesn't fuse as well, and it has a preference, if I can say preference, for a virus for the endosomal route of infection compared with TMPRSS. And so maybe they speculate that changes in the furin cleavage site is causing this, reducing TMPRSS dependence in some way, which is something that you could sort out. Well, but what? So low fusogenic, changed tropism in terms of entering by the endosomal route. What are the implications for what happens in people? And they say, and I think this is really good, we cautioned against extrapolating these in vitro infection data to viral loads in the respiratory tract. Isn't that interesting that they say that? Because some people would say, oh, this clearly explains.
Well, it really fits together nicely because at the top of the paper, they're saying, that how the predictions from sequence analysis didn't necessarily bear out at the epidemiological level. And then they go through this incredibly detailed analysis in culture. And now what does this mean, epidemiologically and clinically? And they're going to say, let's be careful here.
Right.
However, they do speculate that
they do.
The lower lung has less TMPRSS. Right?
Yeah.
No, the lower lung has more TMPRSS.
Has more TMPRSS, right.
So they say maybe Omicron doesn't do as well in the lower lung. It does well, and that would make it. And they say this is consistent with reduced pathogenicity, at least in hamsters, right? Because who knows what's going on in people. So I don't know how you would sort that out in people, right? Because it's, as we said before, it's just so hard to understand the pathogenicity in all different populations. So
you need
you'd need viral isolates from different parts of the lung in infected patients, and you'd need to get all this done before we have a new variant.
But at least in cells and culture, mechanistically, it's quite interesting.
Yes,
right?
It is very interesting.
That here we have a highly successful virus, and then the furin cleavage site changed, and it seems to muck up TMPRSS dependence, but that virus spread really well, didn't it?
Right. So it exacts a fitness cost on the one hand that the virus is not as good in this fusogenic system. It's probably not able to infect cells of the lower lung quite as well. But the net result is that this virus ends up spreading extremely well.
Right. And that may be, it may be that the only thing you need to do that is to be able to evade the existing immunity.
Evade the existing immunity and be in the place where you're going to spread from, which would be the upper respiratory tract.
Yeah. And beyond that, the virus doesn't care whether it, to be anthropomorphic, whether it makes you really sick or not.
Yeah.
So you can lose fitness as long as you gain it in some other way.
Some other kind of fitness,
so the two must have happened at the same time, right? Otherwise it wouldn't work, I suppose. Or else maybe you could gain the fitness and then lose the other.
Well, or if this is happening in an immunocompromised host or in a different species, maybe different rules apply, and you get to the point where you have this form of it that makes the jump.
So that's why, and we have all said this, that's why these experiments are wonderful, because they give you concrete results that are not subject to the same limitations as in people, and you can start to think about what might have happened and make predictions and so forth. I just love this. I'm very glad that we're starting to get these out now.
Yeah.
It's excellent.
Yeah, and the next variant, you can then look at, well, all right, how does it do in these systems, and how is that different, and might that be informing its success?
And all of this is BA1.
Yes.
So I'm sure BA2 is slightly different, right? So how? Perhaps. Maybe it's not. But it is seem to be displacing one in many places, right? So what's going on?
Is there ever going to be a point when these are no longer variants, but they're different, whatever's the next level up?
Strains?
Strains, yeah.
I don't know.
I want to know when we get pi.
Yes, I'm still waiting for pi. I was promised pi, and
well, we passed March 14th, right? That would have been nice on March
14th, yes.
That's a good question, Kathy. I don't know. Who knows? It could happen or maybe not. It depends.
It may be that we get to a phase in the pandemic where we're not naming our variants anymore. I don't know.
Well, I mean, I'm just wondering how many things have to change before you consider something to be a strain and not just a variant. And maybe that's something for the taxonomists, and we'll hear.
So, Jens wrote a long time ago, Jens Kuhn, he said, actually, he wrote this in an article, and he said the designation of a strain has to be done by an international committee of experts, which I think would be tragic, because then it would take years. It would take years. But
anything by committee.
By committee, naming a strain by committee, which...
So we're never getting new strains. Forget it.
But these kinds of this is a different property. But it's not black and white. It's gray, right? It's... it still can use temperus, but not as well,
it is
right?
Clearly different in culture. It does display differences epidemiologically, at least in the populations that we see it in. This is looking like phenotypic difference that I would think of as being a strain difference. But if it takes an international committee, then who knows?
The bottom line is, I don't know what it is, but I can tell you when I see it.
I can tell you when I see it.
I can tell you a strain when I see it, but I don't know how to define it.
I can't define it.
Because is it just in vitro, or does it have to also be in people, or both, right?
Right.
I don't know. Is it enough to be a serotype? So we have 3 serotypes of polio. Are those 3 strains? I think so. They're used interchangeably, but I'm not sure. So, and people listening may say, well, you're a virologist. You should know this, right? Well, we don't.
We don't.
What if one of us decided to take a trip to Canada? Then we would be an international group of virologists.
Ah, yes. We could be an international committee.
So we could then decide on our
Vincent,
own, right?
When's the next CSV meeting?
Yes. I don't know. We missed one already, the Canadian Society for Virology, right? Yeah. So that's where you and I went to, Nova
Halifax,
Scotia, right?
Yeah.
Canadian Society. They're an offshoot of ASV, Kathy. That's what they told us. They were inspired by ASV.
Well, and I'm not sure if he's still the current president, but is Jimmy Dikiakos, and he's on ASV Council. So there's a lot of cross-feeding there.
CSV 2022. Here we go. There is a meeting this year.
Where is it going to be?
Edmonton.
Alberta. Ooh.
Yeah.
When?
Hopefully in the summer.
Yes.
June 5th through 7th. So it's not going to conflict with ASV. That's good. But it was quite small, right? So Jimmy Dikiakos is the president still. Yeah.
Okay. Yeah.
I don't think they rotate every year like ASV. It seems to me that the organizers are similar here. But Edmonton's a lovely place, yeah. Mm-hmm. All right. So, Brienne, you're saying that, Alan, there's a picture of you and me with our 2 guests, and there's a TWiV T-shirt in front on the website for CSV. How cute. There's a rotating banner at the top. And we had a pile of T-shirts, and you and I are at the 2 ends. And I have the exact same shirt on that I have now. You have a purple shirt on. It's CSV-SCV-CA. Yeah. Oh, this is funny. There's another picture of me looking at a poster. Okay. Sorry for the digression, folks.
There it is.
We don't do that very often.
No, we hardly ever digress.
Time to do some picks of the week. What do you have this week, Brienne?
So I have something called the NSERP, or the National Summer Undergraduate Research Project. So this is a project that started during COVID when everything was locked down, but has continued and gotten a bit bigger and gotten some funding, where undergraduates, particularly undergraduates who are from underrepresented communities, can do projects in microbiology, immunology, or cancer biology that are mentored by different scientists. And this is in fact done virtually, and scientists can sign up to be mentors for these undergraduate students, and students can also sign up to be mentors. So this has been, from what I have been told from some of my students who have done it and from colleagues who have been involved, a really useful experience for undergraduates to get research experiences, especially because so many of them have had trouble getting experiences with labs being shut down during the pandemic. And so everything that I know about this is that it's a fabulous program. So if anyone is either looking to be mentored because they are an undergraduate, or if we have any virologists who would be interested in mentoring a student, they should take a look at this and apply.
This is great. And it pays.
And so the students are paid, but they don't have to necessarily go to another location.
Yes.
So they can do it where they are.
Those two things are critical for reaching underrepresented minorities, because two of the biggest reasons that they're underrepresented is being not in the places where these things are made available and not being able to afford to take the entire summer off to go do some unpaid internship.
Right, exactly. So I think this is a fantastic program, and I would encourage anybody who could be on either end of this as a mentor or mentee to take a look.
Yeah.
So they have a link. If you want to make a tax-deductible donation, they have a link. It's great.
Good.
They are a nonprofit. Terrific. I'm going to make a donation after this.
Cool.
I'd like to support this. Cool. Did you say you did this once, Brianne?
I did not, because my lab was shut down, of course, in the summer of 2020, which is when it first happened. But I gave information about it to some of my students, and they were mentees. And had really fantastic experiences, and have basically come back and presented their posters to me of the work that they did.
I love the little saying at the top, #no lab, no problem.
Yeah.
Yeah.
Isn't that great? Love it.
There's a lot of science you can do without having to necessarily pick up a pipette.
Kathy, what do you have for us?
Well, as hinted to earlier, I picked the Virology jobs site, which is part of the ASV site. So you go to asv.org, and across the very top, there's a gray bar, and you can just click on Virology jobs.
Or you go to jobs.asv.org.
Right. And the important thing about this is that anybody can post a job there. You don't have to be a member, and it's free, and anybody can apply for a job that they see there. And there's a lot of different categories. This was put together by Ecropolis, which is a web guru that I know Parasites Without Borders uses, and I think Microbe TV uses. Anyway, it's pretty user-friendly, but if you have issues, you can always write to ASV for help. So that's where I posted my postdoc job. And I just wanted to call your attention to it because there used to be a very different virology job site, and we revamped this when the ASV office moved to University of Michigan several years ago, and I just don't think we really promoted it. So there it is.
Good.
Cool.
Excellent. Great. Yeah, it looks very different. It's nice.
Yeah.
I like it. Rich, what do you have for us?
So I've actually been hurting for TWiV Pics. So I was musing over this a couple of episodes ago, wondering what the heck I was going to pick. And I looked over at my coffee mug and had the same thought that I have, with coffee and so many other things, is that all of the effort and people and time and et cetera that goes into bringing me a cup of coffee or a shirt or, a hamburger or whatever, but let's do coffee. So I looked it up, and I got two sites here. One is 10 Steps from Seed to Cup, okay, which is from the, let me see, it's from the, hang on, here it is, the National Coffee Association, which describes how you get a cup of coffee starting from a seed to make a plant. And it's an interesting process. What really impresses me is to think not only the time involved, but, I look at my cup of coffee and I think that I imagine somebody on some mountainside somewhere, okay, picking the beans that went into my cup of coffee. And that, thank you. I really appreciate that. And the other is a graphic, okay, that I found elsewhere because I wanted a little diagram. This is from a site called Cuptured. And they have a number of different graphics about producing coffee, but there's this one that's 10 steps from shrub to mug. Okay. And are you clicking on this, Kathy?
Yeah, Vincent helped me find it earlier.
Okay. So you get what I was talking about, Kathy, and I've been talking in the background here. The very last bit is step 10 is drinking it. And there's a hand holding a mug upside down. And if I'm not mistaken, that's a University of Michigan logo on the mug.
Oh, yeah. But I'm almost immune to it now. Like, I'll see it in a weird place. I remember seeing it someplace at a very small locale kind of graffiti in Europe. And then suddenly it hit me, oh my gosh, this is Michigan and I'm not there. Yeah.
So when the mug is upside down there, is that Wisconsin or are the colors wrong?
No, their W is different shape.
It's so red, right?
Okay. At any rate, so, people might find this amusing and, think about it with gratitude for the people who bring you this stuff and don't waste it, All
right.
If you throw the dregs of your coffee down the sink, somebody's hard work is going down the drain.
It's interesting you pick this because this is the first thing I do in the morning, right? When I get up, I go right to the coffee and I put the water in and I grind the beans and then I go walk the dogs. They got neck. They don't go first. They have to. And it's comforting just to do this, right?
Yeah.
And it smells great when you grind the beans. And I figured out what coffee I like and how many scoops to put in and how long to grind them. It's just, and then when I travel and I don't have this, it's like the mornings are, what's going on here?
Well, this can be part of your meditation, is to think about everything that went into bringing those beans to your kitchen.
I do. I feel badly looking at this bag, right, of beans, and the bag gets thrown away because it's not the recyclable paper. And I'm wondering, where this came from and how much water was used for this. Yeah, I think of that a lot.
It's amazing how much the pandemic and the everything shortage has gotten us all thinking about where stuff comes from.
Yeah, for sure. And somewhere someone told me how many gallons of water it takes to make one almond. It's just crazy.
Yeah, it's so much.
That's another one.
It doesn't compare to the number of gallons of water you need to make one hamburger, though.
There's another statistic from this site that I need to mention, and that is, by my calculation, this was the predicted coffee bean production for, this is unroasted but dehusked, ready to roast. Coffee bean production for this is 2015, 2016, so it's probably higher now, was, by my calculation, 20 billion pounds. So that's 3 pounds of coffee for everybody on the planet, right? So somebody out there is not getting their share because I'm doing more than 3 pounds.
Well, a huge proportion of the world, it drinks tea.
Yeah, it's true.
And in the tea-drinking countries, coffee cannot gain a foothold.
I'll bet you I do at least a pound a month and more like 2.
Okay.
So it takes 37 gallons of water to make the beans needed for a cup of coffee.
Wow.
That's a lot of water,
That's a lot
isn't
of water.
Oh, cool. Alan, what do you have for us?
I have a book that I just read recently. Came out in 2020. It is called Fathoms, The World in the Whale, by Rebecca Giggs. And this is a pretty amazing book. It's I picked it up because I saw a couple of good reviews of it, and I thought, wow, whales, that's a Massachusetts thing, and I know a little bit about them, but not really. And she got interested in whales, and she's a journalist, and just dug deeper and deeper into the subject. And excuse any apparent puns in this discussion because it's impossible to avoid them. It is a huge subject. And just the biology of them, the cultural significance of them throughout history, the history of whaling, which was some stuff I knew, but a whole lot of stuff I didn't know. That whaling actually ramped up a lot during the 20th century and when we didn't need the whale oil so much. And the whole thing with whale falls, when a whale dies in the ocean and the carcass goes underwater, it takes a very, very long time to reach the bottom of the deeper parts of the ocean. And when it does end up there, there's a whole ecosystem that blossoms around decomposing this that goes on for years. There are organisms that only do this. They decompose whale fall. And all this is in there, and it's so that's all cool factually, but a lot of science books, and I'm used to this, and I like it. You're reading a science book, and you want to hear the facts, and you want to hear them interestingly presented in interviews with the researchers. She does some of that, but her writing is just so gloriously intricate. I mean, it's almost like poetry the way she writes, and putting these enormous animals into this broader context. It's just the kind of book that you read a little bit and set it aside and think about, and then you read a little bit more and do the same. Like the whale, it is something that you need to take a piece at a time.
Cool.
That sounds really interesting.
Yeah.
I'm looking at a little selection here. You can read her writing. Yeah.
Yeah.
Her sentences are long.
Yeah, and extraordinarily well constructed. I mean, as I admire this as a writer as well as a science journalist. It's amazing work.
So, Alan, when the whales wash up on the beach, did something go wrong that they didn't sink?
Usually that's the result of, so when you hear about these whale strandings, it's the result of something going wrong and the whale got sick, or the whale got hit with Navy sonar and got confused, or they freaked out for some reason and they ran themselves up. It's poorly understood. But when these things happen, they often happen in clusters where, a pod will go and do this.
Wow.
So you have to check out today's astronomy picture of the day, which is an aurora shaped like a whale.
Oh, cool. That's neat.
Good timing. I did not know that.
Sweden. Nice.
Oh, yeah. I need to put them back in my RSS feeds. I don't know.
I'll tell you, they are now my everyday, thanks to Kathy.
I give Kathy a lot of credit because every time I open a web browser, that's what I get, and it makes me happy every time.
You saw that one yesterday of the hourglass shaped thing. That's the summary of days.
Yep.
I can't describe it. It would be a whole nother pick.
Cool. All right. Well, speaking of water, my pick is today is World Water Day, and there is a website, worldwaterday.org, and it's all about groundwater. Groundwater is invisible under our feet, and it's going away. So we need to work to try and sustain it. So, I mean, this obviously the day is about awareness, right? That's today. But the website has some things that are interesting. They have some reports from the UN, et cetera. They also are running a little contest where you could make a 1-minute video about, why water is important. And they have some other information and so forth, posters. But it's something you should learn about and realize that we have to be careful. Or it's going to go away, and it sustains life. It's really important.
Yeah.
Cool.
World Water Day. Every day is someday, but that's good.
Yes, that is a very important one.
We have a listener pick from Jeremy. Greetings, TWiV team. Wanted to share this video after your recent discussion on the universe. It's a virtual tour of the universe from the EPFL Astrophysics Lab in Switzerland. It's called Archaeology of Light, an immersive journey through space and time. It's very cool. Lots of good graphics here. The highlights for me were the simulation of the galaxy spinning almost like a hurricane, the complex structure of galaxies that make up the cosmic web, and the graphic showing how our radio signals have barely reached a fraction of our own galaxy. Thanks for all you do, Jeremy. So thanks for that, Jeremy. And that will do it for TWiV 879. You can find the show notes at microbe.tv/TWiV. You can send us your questions, comments, pics to twiv@microbe.tv. And if you like what we do, consider supporting us. We depend on your support to do all of this. Go to microbe.tv/contribute. We are a nonprofit, so your contributions are tax deductible.
In the
Kathy,
U.S.
In the U.S. I don't know what happens elsewhere. Do they have similar structures elsewhere?
They
Alan?
do, but I don't know what happens if you're a Brit and you give to a U.S. charity. That's
Don't know.
Nothing we say on TWiV constitutes tax advice. Contact your tax professional.
That's right. Kathy
Past
Spindler's
performance is no indication of future results.
Kathy Spindler's at the University of Michigan. Thanks, Kathy.
Thanks. This is a lot of fun.
And if you didn't notice, Kathy has a new background today.
It's just a one-time deal. My office is getting recarpeted, so I'm in the departmental conference room.
Alan Dove's at alandove.com. Alan Dove on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
And Rich Conda is an emeritus professor at the University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.